This talk is a modification of a talk that I recorded for NVIDIA GPU Technology Conference, which is going live next week. So if you like some of what you see here and you're interested in the kind of what GPUs can do, it's free to register. A big international conference runs, I think, from the 12th to the 16th. And there's some really interesting looking talks. So just a little plug there. But today I'm going to be talking about, as Peter said, portable sequencing and how it can disrupt and empower, and tagging in the GPU side of things just because I'm a massive nerd. So for those of you that have heard me talk, a lot of this is going to be familiar, so you can tune out, do whatever. I will highlight when there is some cutting edge new stuff in there. So I was saying to Peter before, I actually stitched some results in about 10 minutes before this that I think are quite exciting. So let's go. The talk is, all of my talks I try and all my code is freely available. I paste it into chat right at the start, so it's there and we can paste it again. The recording will be made available as well. So everything is freely accessible. So there's actually a lot of documentation that underlies a lot of these slides that is potentially interesting to people. So please feel free to follow along or find it later and have a look. So Peter mentioned that I have affiliations with projects. I'm also on the Bioinformatics leadership team with Mick, Kim and Rudy for Genomics Aotearoa. And Leah is one of the researchers that we have in a research position as well with the FTE. And so I'm going to talk about some of the work that she's been doing too. So hopefully a bit for everybody. So, I mean, this talk, as I said, has been modified from the NVIDIA talk. So I think most people are pretty happy with who we are at ESR and who these lovely people are. And sorry, I forgot to include Gemma in here as well. But you know, the genomic response, or the response that we've had as a nation to COVID, including the genomics, has really spurred interest in genomics, which it's quite cool to be in a field that is actually seen as sexy again. So it's really pushed a lot of this stuff along. And so I should have included the picture of Yoop in his paisley shirt with Jacinda, but this, I think everybody's fairly happy with ESR. We do genomics, we do a whole lot of other stuff, surveillance. I think everybody's probably happy with genetics and sequencing. So I'll skip through here. I'm part of the Human Genomics, so I'm the Bioinformatics lead of the Human Genomics group at ESR, and we do a whole lot of independent research. So Donya McCartney-Coxon's the lead of our group, and the bulk of her research concentrates on type 2 diabetes and obesity. We do cool work with Max Berry from the neonatal unit and from Otago. I'm going to actually touch on that as well later from a different aspect, but we do work with the guinea pigs, and we're really passionate about empowering people and letting them explore and interpret their data. One of the GA projects that I'm helping out on part of is Stephen Robinson's clinical diagnosis through genetics, so using whole genomes to upskill the DHBs. And we will talk about that a little bit and some of the work coming from that. And then I have some internal funding in collaboration with Richard Carroll, looking at exome sequencing for hyperparathyroidism patients. We're just getting the sequencing underway for that. So there's some exciting stuff going on. So we do a lot of sequencing. Traditionally, we've done a lot of that sequencing on these beasts, these very big, very expensive, very monopoly-orientated Illumina machines. So I'm not going to be talking a lot about those guys after a certain point, but this is where we think of next-gen sequencing. Most of us probably think of Illumina-based short reads, expensive machines, rather large, highly accurate. And they're kind of the mainstay of most genomics until probably the turning point in the last couple of years. And so we've probably all seen this as well. I just like the quote. You sent me this slide, so I stole it off him. But I really like the quote that it's soon going to cost less to sequence a genome than it is to flush a toilet. And I mean, that's the thing, right? It's getting cheaper and cheaper to sequence, and it's becoming the compute and the informatics that's the bottleneck. So that really ties into how we use technology to speed things up. And now we get into the meat of what the nerd and the geek in me loves is the technology here. And I'm so lucky that I get to bridge my love of computers with biology and things. It's a really humbling place to be. So we started to get into the GPU scene at ESR and started accelerating things. And it wasn't until I was at eResearch in Dunedin last year, 2020 in February, when you could still do in-person conferences. And NVIDIA had a presence there, and I was in a really dry workshop where they were talking about even stuff that bored me in terms of the low down architecture of GPUs. But they mentioned this company that they just bought called Parabricks and how they were doing these crazy things to speed up genetic pipelines. And so it piqued my interest, and I managed to get a trial license. And so what Parabricks does is they've essentially taken all of the various open source tools that we're familiar with, things like BWA, GATK, Star Aligner, various Mutex, CMV Kit, Deep Variant, all of these amazing tools. And where they weren't previously able to be accelerated by GPUs, they've torn them down and built them back up to be sped up by GPUs. So GPUs, graphics processing units, I should probably touch on for those that aren't that familiar. If you've got friends, family, children that are begging you to buy graphics cards to speed up their Fortnite addiction, then it's the same technology. They kind of existed in the realm of graphics, and over the last kind of 10, 15 years, they've become very good and very able at parallel processing. So GPUs are really good at taking a single job, breaking it up, and doing it very fast. And so that's what was done with these kind of gold standard bioinformatics pipelines and wrapped into Parabricks. So does that matter? Yeah, it actually does. So with our exome sequencing, 100 times coverage exome takes between 8 to 12 hours on one of our Linux servers. It's at 3248 cores. A 30 times genome takes 30 to 40 hours. For reference, these two V100 GPUs can do 100 times exome. And what I've been here is from raw fast Q data to VCF. So we can now do 100 times coverage exomes in 6 to 12 minutes on the two V100s. And then 100 times Illumina Platinum genome can be done in roughly 4 hours, and that's on two V100s. And so these things scale, and Illumina's claim is that when you've got 8 A100s, you can run a whole Platinum genome in about 20 minutes. So that's from raw fast Q data to VCF, so variant called format, in 20 minutes. And that blew my mind at the time. I didn't believe the claims, but now that we've actually got these things in our hands and we're doing it, it still seems a bit like black magic to me, but it's really exciting. And it's really exciting in terms of how we can potentially clinically and rapidly turn things around. So from our end, the bioinformatics no longer is the issue. We're still using Illumina sequencing. So those sequencing runs are upwards of 30, 48 hours to actually generate the sequence data before you can analyze it. And so, we've got that lag time, and then you've got the time for clinical interpretation. So the compute is essentially solved, in air quotes, in terms of how quickly we can do it. It's really exciting. Parabricks is also growing. So we jumped on, it must have been at the start of 2020 when it was quite bare bones. And now there's lots and lots of other pipelines making their way in, a lot of long read based stuff as well. So you got like Benito, Recon, Medaka, Raven. So a whole lot of tools that you're probably used to are being incorporated. And what I forgot to mention is that these pipelines, you can run a single pipeline. So one line of code will take your FASTQ files through to VCF and everything in between. But you can also break down that pipeline so you can use the isolated BWA that's accelerated by GPU. So if you just want fast mapping, you can use BWA that's GPU accelerated. You don't have to be running pipelines. You can actually use the individual tools as part of this. And so we've been pretty happy. I mean, we've just signed on for a second year in terms of licensing. And so Parabricks, it does have a cost, and that cost is $2,500 US for commercial. Well, yeah, ESR's kind of, we pay the expensive license. I think education license you can get a discount. So it's $2,500 per GPU. So there's a little bit of money there, but it's not huge. Denny and I were having a chat the other day about how we feel about people taking open source tools and charging money for them. I still think it's at a price point and it's transparent enough, and the results that we're getting are probably good enough. But I agree it's a little concerned, but I'm still excited by where this is going. And as such, Nessie's actually just gotten a lot of the A100s in. Doesn't look like this beast here, but it's awesome to see that we're really pushing GPUs now, and we've been working closely with Denny. Leah, who I mentioned before, in fact, has been working with Denny, and just last night was running a genome on the two A100s on one of the nodes on Nessie, and it's really cool that I get to work with like-minded people whose idea of fun is just crunching and watching these things really thrash themselves to generate data really quickly. And so Leah's been instrumental since she's been on for just over a year now, and she's taken a lot of our pipelines and really made them quite beautiful and added immaculate documentation to them. So these are all links out to the GitHub version of these pipelines that you can feel free to have a look at and even implement. They should fairly readily mesh into most systems, be it servers, computers, high performance compute clusters. Leah's implemented everything in SnakeMake, and I think there's some Slurm wrappers there as well. And they're also CPU and or GPU aware where available. So she's done some amazing work on that front, and it's awesome to see where we're pushing it. And we are pushing it. We're working on some of the clinical pipelines to move from the SNP and indel calling into things like structural variants and CNVs using some of the tools that are built into deep variant. So I'll come back in another 6 months or so and have an update on that, but it's really exciting. So that's all I want to talk about in terms of old school technology. And I want to talk about another type of technology, and it's not going to be PacBio. So that leaves us with Nanopore. And so most people are probably familiar with Nanopore. It's a long read technology. I'm definitely not shilling. I'm trying not to fanboy, but I am a huge fan of the technology. And so in my mind, is a really elegant mesh of biology and technology. And so it's a mix of, these biological proteins and creating pores in a membrane, and then pulling, these strands of DNA using motor proteins and helicases through, having an electrical current across there, and each base disturbs that current in a unique signal. And then we can turn that signal back into sequence data. And I think that it's just completely mind-boggling at the scale and the fact that we're talking about long read data here. And by long read, if you're used to Illumina data, you're kind of talking in the hundreds of base pair range. If you're not that familiar with Nanopore, you routinely get 10 KB, fairly easy these days, so 10,000 base pairs. But with various kits, circulomics, ultra long read kits, small read eliminated kits, you can really push that. And now people are actually routinely getting in the hundreds of KB, so hundreds of thousands of bases. And then there's the whales, the reads that are just so massive that it's ridiculous. I think the current world record read is about 4.2 or 4.5 million bases. That beast spent 3 hours going through one poor little tiny pore. 3 hours of time occupancy to sequence that read at that length, but it's just amazing. I embedded a video. I could play it quickly, just because it's quite beautiful. I'll skip past. So the MinION's a little USB-sized device if you haven't seen it, and your DNA gets prepped with adapters, a motor protein, and loaded. So that little part there has got the membrane with the pores on it. And then physics works its magic, and this stuff kind of slowly makes its way towards these pores, and then you have tether proteins that will locate them, as you'll see coming up. And so they get located, and then with an ATP-based reaction, the motor protein and the helicase unwind and pass the strand through the pore. I could look at this all day. It's beautiful. It's just incredible piece of, as I say, merger biology and technology. And then there's electrical signal across, there's a potential across this membrane. And as the bases pass through, you get this disturbing current in these squiggle plots, we call them, or peaks. And so I'll leave that for you to finish off later if you want to, but this is an example of a tiny section of a squiggle plot, and you can see the representation of the bases. And what I should also allude to is, other than the long read aspect, there are other benefits to the long read technology. One being that if you're doing a fairly basic genomic prep and not doing any kind of amplification, you will also receive base modification information. So things like DNA methylation. So I think the base caller Guppy, the software that turns the squiggle into actual sequence data, FastQs, at the moment by default can call 5' methylation, 5-hydroxymethylation, and 6N adenylation, or 6N methylation of adenosines. And there's some beta software with various other modifications. And so this is really exciting because we're starting to find more and more potential modifications, and the fact that these peaks can differ. So a methylated cytosine peak differs from a cytosine peak, and that can be picked up. And so traditionally on CPUs, that process actually takes quite a long time. But GPUs are actually really good at taking that type of data and processing it really fast. And so one of the examples I like to use is when I first started I'm pretty new to Nanopore, and terms, they've been around since about 2014. I think they really hit the scene. I think I started when I started at ESR because somebody came to me and said, "Oh, we run this MinION. It's taking like days and days and days calling on the laptop, and it would have taken about 7 days calling on the laptop CPU. So converting those squiggles into bases, and I was lucky enough to have a shiny new toy on my desk essentially when I arrived at my job at ESR, and we crunched that data in about 7 minutes. To put that into perspective, so the speed up is ridiculous with these things. One of the things I might stave off a few questions is that there's always the cry of accuracy. It's becoming less and less of a problem. The chemistry is getting better and better, and the software, the algorithms are getting better and better. So I mean, that's one of the things that's amazing is that we can be using a MinION, one of these things here. You can see my camera or not. That we got in early access in say 2015. That piece of hardware is still completely viable. It's the software and it's the reagents that are changing to improve that accuracy. So when it debuted back then, it might have been in the 70s and 80s. Accuracy now is getting up to, with particular callers and particular flow cell chemistries, 99.9, 99.1. And you've got to remember that's across reads that are thousands or hundreds of thousands, if not millions of bases long. So it's a really exciting time, and it's only going to get better in terms of that accuracy, and it has only gotten better. And you've got polishes and things. So software can solve a lot. So I've spoken about this. GPUs can do things really quickly. They implement things called neural networks. I'm not going to delve into technical detail here. I just want to kind of give a fun story time overview of this kind of stuff. But they have lots of things called CUDA cores that allow you to split your job up, run it across all the CUDA cores, bring it all together at the end. So we've been doing some benchmarking. I need to update this soon. We've just got 2 A100s ourselves, so they're the successors to the V100s. So we're going to be updating our benchmarking link soon, but that link's there if people are interested. So we'll step back into story time again, and this is a kind of personal story. So this is my son, Jamie. He was born 32 weeks, so eight weeks prem, 2 months prem, and that's him there probably a couple of days in. That's my giant hand. And then this is him when he was nearly three years old. So I just wanted to say he's fine, he's healthy, he's absolutely okay. A couple of days in, maybe 9 days into the period when he shouldn't have been born, he had to have a lumbar puncture because he spiked a huge temperature, and they didn't tell us, but they suspected viral bacterial meningitis. That test took more than 48 hours to return. And so me sitting there, why can we not use all of this technology we've got on hand and have it as close to point of care as possible? And so you can imagine a situation where you can then take a sample, blood, saliva, nasogastral, cerebral spinal fluid, you can extract the genetic material, you can prep your library, you can get it onto the machine. And the thing with Nanopore, I mentioned before that you have that lag time with Illumina before you can analyze the data. Nanopore doesn't have that. As soon as the DNA is coming through the pores, you can have that sequence information. So as soon as you see a piece of virus, a piece of bacteria that matches, you can start to ID these things, and then you get more confidence as you get more reads that cover that. And so, the time to first read match detection could easily be within an hour. Within a couple of hours, you could even be closing whole genomes of particular viruses and bacteria. And this isn't imagination anymore, really. This is pretty soon to be reality. And so that got me thinking, and I then came across some other technology that helped me put the pieces together. And so one of the aims was, can we make it cheaper? And the good thing is that mobile phones just seem to be getting more and more expensive. It's ludicrous to me that people can pay $2,000 for a cell phone. So we can easily get compute that can run these sequences for less than that, making it accessible. It's small enough to be portable. I may have performed an inadvertent drop test while there were 7 or 8 ministry people in the office. The thing dropped from my standing desk about a meter and kept going. Nothing missed a beat. So that's just a spoiler for when we get to the technology. And then you can imagine the use cases are really just bounded by your imagination. And the more and more people I talk to, they go, "Oh, have you thought of this?" I go, "No, that's awesome." So, and I'm really excited in terms of, the clinical setting and field, the portability to go into the field, but also the ability to not just go into the community and to do teaching and things, but also to even take that gear and leave it there, or say, here's the recipe to create this yourself. And then you're teaching people about not just the technology, but also genomics as well. And then you're spurring that wave of people that might not have had access to that kind of information. And I think only good things can happen. So this was our experience. So I like to keep up with technology, and I've got a few Raspberry Pis at home doing various things, and I always thought it would be cool if they had decent GPUs. And then a few years ago I stumbled onto these boards that NVIDIA were making. So these are essentially Raspberry Pi like in terms of they're a single board computer, but they have beefy NVIDIA based GPUs on them. So this boy here is a Xavier AGX. So this is 8-core ARM processor, 16 gig of RAM, and a 512 CUDA core GPU with 64 tensor cores. This comes in, so this in context will be more powerful by a decent mile than most of your laptops or desktop computers. And it costs 1,000 New Zealand dollars. And so we got one of these and a few smaller ones in, and I was able to get base cooling working after emailing ONT once a week for 9 months until they decided they couldn't get rid of me and they wanted to help in the end. So we were able to get ARM builds of Guppy, and so we started base cooling. And so that was really cool. And you can find some of the documentation and notes up at that link up there. So benchmarking, what I just wanted to see is you could actually put the Xavier into various power states. So at max, it's drawing 30 watts, which is probably less than twice what most laptops will pull. And at full power, it would do that same data set I spoke about before in 7.5 minutes. So it was actually pretty on par with a $5,000 GPU that we were using, which blew my mind. And then as you cut the number of cores and cut the amount of power, you can see it does take longer. But compared to, say, days and days of base cooling, you're still only talking about half an hour down at 10 watt mode, which is just incredible. And so this is just a rough, this is mainly more for the graphics card people at the conference next week, but this is just a slightly outdated now comparison of the kind of performance of these various GPUs and Jetson boards, their price point. And it's quite impressive when you see the Jetson Xavier pulling 11 teraflops at $699 for 10 to 30 watts, compared to cards that are, it's not a lot more teraflops than 16. It's not a lot less teraflops than 16, but it's a lot less power and price. So they're quite an enticing little option. And so I started to get more and more into GPUs from the compute and the science sense. I'd always been into them from what they were doing for the design and the gaming industry. And so people started to reach out because, we were quite transparent with blogging and Twittering and, Redditing and all of those good things about this stuff. And so Jean Barker reached out to me on Twitter and said, hey, he works in a bioinformatics institute looking at cattle viruses in Nairobi in Kenya. And he's like, hey, we're doing a lot of nanopore stuff. I want to buy one of these devices and potentially get it portable. And my caveat at that point in time was, well, we haven't actually got the MinION running. The software stack's proving difficult because they've got different compute architecture. We're going to get there, but I can't guarantee we're going to get there quickly. So have you thought about actually just buying a GPU to speed up the process? And he said, well, yeah, but IT said we've got to buy $8,000 ones, blah, blah, blah. And I said, well, I don't know what the situation's like in Nairobi. I don't know how easy it's to go buy a gaming card from a store or from Amazon. And he said, oh, I'll see what I can do. Within a couple of days, he sent me some pictures. And he's like, oh, yeah, I've got this NVIDIA GeForce 2070. So a fairly back in, about a year and a half ago, it was a fairly decent gaming card. And he's like, cool. So I'm going to install it in my desktop server. And I was like, awesome. And then he said, next problem, this thing's got an 8-pin power connector. I've got 2 6s. I said, again, I don't know what your electronic shops are like and if you've got access to Amazon, because you can buy a converter for about $15. He said, no, that's not going to work. And so a few hours later, he had spliced 2 off one of the 6s and put them onto the other 6 and plugged it all in and was happily base calling. And so they were taking data that was taking them weeks to base call on CPUs, and they were doing it within hours, which has really sped up their time to detection of viruses in their livestock and things, which, it was really awesome and humbling to be a small part of that. All I did was say, hey, you could get a GPU and you could plug it in. And now we're actually looking at writing a paper together about GPU compute and MinION data and things. So it's really cool, the doors that it's opened. And it's opened many doors. So I mentioned Jean Barker. John was in the piece early. He'd bought a couple of Xaviers and was following along with my posts. Luigi was kind of doing this in tandem, and he got it working on an older version of the Jetson board. So we were swapping notes frequently. So he was in Italy. Jürgen, who I'm going to touch on a bit, has been incredible. So he's a MD, PhD at Basel Neuro Hospital. And so he's got a vision of actually getting these things into the surgery and doing, biopsies and actually live sequencing while the operation's still occurring. And it's just awesome. So him and Luigi have been an incredible part of us troubleshooting the software issue and getting it up and running. And then just before Christmas, NVIDIA actually got wind of what we were doing and were really excited. And so I actually meet with them fortnightly now. And they're really helping us speed up software development because they're able to put their sizable might and money into converting architectures into, from x86, 84, so more traditional CPU-based architectures into ARM, which is on the board. And so, and also more recently, ONT, I'm having frequent meetings with Oxford Nanopore, which has been really cool. And so this is unfolded as a story on this GitHub gist. So if you have a spare hour and a half or so, there's a really interesting post that go from when we just had the Xaviers doing nothing to 3 a.m. troubleshooting because my son was awake at that hour and Jürgen was awake, and excitement because he got base calling working for the first time. And so it's just this really cool cool vibe in that thread, and some really cool information in there if you're interested in that kind of thing. So back then I was thinking, this is the future, these little boards. And I mean, these little boards are used in industry and everywhere now around the world. Like, they're doing all sorts of cool things, like autonomous cars have multiple Xaviers in them. So they're by no means a niche case for what we're doing here. We're just using them for genomics. The not so distant future was maybe 7 or 8 months ago. I was like, well, this is where we want to be. At this stage, the MinION in these pictures is just for show because I didn't have the software stack running, and there's a dollar coin. So this is a portable power bank that would easily power this little board. So this is a $600 Jetson Xavier. So this is the NX, 6 cores, 8 gig of RAM, and 384 CUDA core, 48 tensor core GPU for those that like that kind of stuff, the stats. So $600 plus a terabyte hard drive, NVMe drive would be another couple $100. So for $800, you've got this compute. And then, a few $100 for a battery, another little hard drive, and, MinION comes free with reagents these days. So that setup is really cheap. And so that's where we were wanting to go. And sorry, I can't remember if I mentioned dollar coin for scale, so you can kind of see the size of these things. Where we actually are now is my dining room table here, much to my wife's dismay, the amount of things that have happened on that dining room table in terms of sequencing various bits and pieces. But so this is the software up and running. So we finally cracked that nut, and we've got the full Oxford Nanopore suite of software running on the Jetson boards. And so this came during lockdown, the big lockdown last year, the Jetson Xavier, and I think it was probably towards this time. No, it must have been. It must have been maybe nine or so months ago. We were up and running, and this is a picture of Matt's hand putting COVID onto the MinION on our desk and getting up and sequencing. And then this is a kind of setup with a solar panel as well, because you imagine, and we have taken this thing portable. And so we can also pair, if you don't need the portability, then all I said, as I said before, $600 for the compute, $200 for the hard drive that goes in it, 24-inch touchscreen, and this is a MinION sequencing platform. It will live base call in fast mode, and we actually use this in Christchurch. We have been for a few months now to sequence COVID, doing COVID runs. This setup here at eResearch back in February this year, I had the setup in my talk sequencing COVID live, and this is the Rampart dashboard that runs on the board. So if you stop and think about that, that's running the MinION, doing live sequencing, live base calling, and serving up Rampart all on a machine that's $600 is pretty ridiculous and incredible. We're 3D printing cases for them, and I've got a little army in the door about a month or so ago, and I've got some other cool projects that I'm not quite ready to talk about yet, but yeah, we will be talking about that. And this I couldn't resist to put in. So I haven't spoken about them, but they're the ones, the cute little $100 boards. So the Jetson Nanos. So they've got 4 cores, 4 gigs of RAM, 128 CUDA core GPU. And so that was my original vision nearly 2 years ago, is let's get that thing because it's $100. But there's various hardware limitations that mean it can't, caveat, it won't easily base call. But if you're not worried about live GPU base calling, it will happily sequence. And we've sequenced SARS-CoV-2 on this setup. So a $100 compute can power a MinION. And so I didn't mention it before, but in that picture of the ONT hardware, there's the smidgen, and that's always the joke that it's vaporware. If what we're seeing on this type of hardware for $100 is true, I don't think it's going to be too long until we see nanopore smidgens on cell phones. It's awesome. Yeah, what you can do with this tech with such little compute power is incredible. And so one of the projects that we're really excited about is working with Georgia Bell from ESR in Christchurch, and she's got some great partnerships with Māori and iwi down there. And we've got a project to look at the health of particular waterways. And so along with Matt Story, who's working on all the extraction protocols, our aim is to take the setup to the source and actually do the DNA extraction, the library preps, and the sequencing. So we've sold the sequencing side in terms of its portability and its power. And Matt's currently in the lab working on the extraction protocols. So once that extraction protocol is set in the lab, we'll take it out into the field, and hopefully again in 4 or 5 months' time, we might be able to report back on that. And it's awesome. And it means that you can do that with community members, and building on that and showing them that this stuff isn't black magic, and it's not incredibly difficult. I believe with some proper training, even I could probably do the wet lab work required, and I haven't been in the wet lab for many years. So I think it's just what you can do and what we can offer in terms of the experience and the teaching and the empowerment is amazing. And so hot off the press, a few people out there probably heard about read until or adaptive sampling. For those that haven't, I'll spend a little bit of time on it just because I think it's an amazing piece of technology. So remember that we're sequencing by passing DNA through pores. And so when you've got a GPU, you can do this, as I mentioned, in real time in terms of what's come through the pore. You can actually be turning that into sequence data and aligning that against references, right? So you can be searching for that and trying to decide what that read is before it's even come fully through the pore. By utilizing that, if you take the first 450 bases, the current rate of transfer of a piece of DNA through the pore is about 450 bases a second. With that first 450 bases, you can have an ID on that read, and then you can actually decide if you want to keep it, in which case it'll just come through the pore in sequence, or you can actually reverse the polarity and eject it out of the pore. So you can start to essentially enrich and or deplete for things that you want or do not want to see, which is incredibly exciting. And I'll talk more about this with the stuff that's hot off the press in terms of an hour or so ago soon. But I'll come back to Jürgen, and he got hold of me, it must have been about 2 or 3 months ago now, when we were really steamrolling through, once we got the software thing solved, we were like, can we get adaptive sampling running on the Xaviers? Because, that was really exciting to know. And he got it running on the Xavier on a cancer sample and was able to actually type a cancer. They'd already typed it with other sequencing, like with other long read sequencing, but as a proof of concept, they typed it on the Xavier setup with adaptive sampling running, using methylation alongside SNPs. And that was really exciting. And so we're in the process of putting a paper together on that as well. And if I remember the segue, I'll touch on why there's a really cool little bit of biology around reads that you would usually throw away there. And so this is literally data that was generated a few hours ago. So this is what it looks like when adaptive sampling is running properly. And so you remember me saying that when you're running this, you're looking at the first few hundred bases that come through the pore and then making a decision. And if the thing that you're enriching for isn't a large portion of your sample, you can then imagine that the bulk of your reads are going to get rejected. And so that's what we see. And all being equal, should be able to jump into my rough notes. So I will tidy this up. I was just too embarrassed. It's private at the moment, but I will tidy this up, and it will become part of the talk through the links that you've given. So what we did, Matt Story here at ESR yesterday prepped up one of the Zymo metagenomic communities that we have. So this is just a breakdown. So 8 various bacteria and a couple of yeast species in there. And we thought, well, let's give this a whirl in terms of let's try to enrich for, and I didn't look at the table here, which was good. It's a good first step as a scientist to not look at any of the data, but I just thought this has got the biggest genome, this yeast that I hadn't heard of. Let's see if we can enrich for that. And so this is the run, this is without adaptive sampling, so to kind of get a ground truth. And so it looks like this Zymo kit, and it's been freeze thawed a lot, so it's no surprise that it is quite fragmented, so our N50s aren't large. But we thought there's still probably enough information there. And so we ran it for half an hour, 40 minutes, and you get a breakdown. You run it through Kraken, and you kind of get what you expect. The unclassified, the Kraken database I had didn't have yeast in it, so all the yeast reads are in the unclassified reads. When I turned on adaptive sampling, and remember we're looking to classify this yeast that's actually in a very, very small amount of the total data, the total DNA pool, here is that peak again that you saw in the previous slide. Here it is blowing up. And there's a lot of megabases of sequence that's been thrown out, which is what you expect, right? Especially probably doubly so because that yeast species is in such low level in the total amount of DNA. What we saw though was essentially in terms of, and what's important here is the actual read count, don't worry about the percentage totals, a nearly 5-fold enrichment of that particular yeast species. And it was 90, I think it was about 97% of these reads all mapped to that particular yeast species. So that was incredible, and that's really exciting. I was talking to Yoop about it, and one of the things that a lot of us here, and I know a lot of people are asking and passionate about is that point of care. And if you can identify those things that are floating around at low levels, that is really the goal. And so that was really exciting. That was yesterday. Today I had to play with depletion. I was like, well, if I take some of those bacteria that are really present and our yeast strain that wasn't super present but has a big genome, what if I try to knock those down? And so I got an interesting curve, and I need more time to sit and to play and to think, but you still get this big bulk of reads getting thrown out, but then you've got a lot of reads coming through, which I think makes sense. And I ran it through and I was like, oh, okay, everything's there. But then again, you and I were talking about it, and I was like, Miles, you've been an idiot, because of course these reads still come through the FASTQ file. So you end up with a whole lot of short reads in your FASTQs, and for Kraken, that was enough. It was more than enough to make IDs. So if you get rid of reads that, and I picked 1,000 base pairs, I said, if I get rid of reads that are 1,000 base pairs or less and run Kraken again, none of those 3 species that I was depleting come through at all. And to me, that is awesome. That was really exciting. And then yesterday, we were also seeing if we could do this kind of thing on the Xavier. So you get a very similar profile to what we saw on our server on the Xavier. Where it falls down is it's working, the adaptive sampling is working, but we don't think the CPU is actually fast enough to keep up. So you see this lag where we expect this peak of reads getting chucked out at around 500 bases. It's shifted up to around 2.5, nearly 3,000 bases. Still some interesting information. All of the reads that are passing at a certain length, the bulk of them are mapping to the yeast species. And what was really neat is that the longest read out of any of the runs was on the Xavier, and it's got a really nice query and blast in terms of it was a nice read. And this was only using the fast model, so that was really exciting. And we were playing on the grid. I'm not going to go down into my messy notes down there. We were playing on the gridiron, and I need to go back and have a look at that, because adaptive sampling is built into the gridiron, but for some reason we weren't seeing the same enrichment that we were getting off one of our custom servers. So it was interesting. So that's literally hot off the press. So I'm happy to hear people's thoughts and talk more about that. Not so much hot off the press, but still cool, is the fact that you can now download the Minnow app and control these things from your phone or your tablet. So you don't actually need to invest in like a touchscreen or a portable screen. You can deploy this thing as long as it's got an IP address and a network connection. You can actually log into it and start, stop, control, view your sequencing runs from a mobile device or a tablet or even another laptop. And that's really neat. So I'll start to wrap up and hopefully have a bit of time for discussion and questions and things. But we've been documenting a lot of this. Some of it's been getting some press. We had a little bit at the end of the really great article that was published in the Herald. Unfortunately, it's still behind a paywall, so we've got a similar story to the bit that we had up on the ESR website. It's for those of you at this talk, it's pretty much what I've talked about in terms of the journey and like the where it came from and where we're headed. There's a repo, a Git repo, and I think I've got enough time to open this up. So this is essentially all of my notes, including components list and setup code and stuff. So you can actually do this thing yourself. Pictures, because I love pictures of unboxing stuff. I don't do videos, I just take pictures of hardware. So for example, this is nice. It kind of breaks down that picture we had before where if I do some quick maths in my head, I think it's somewhere around the $1,300, $1,400, $1,500. You can have this setup that you can go anywhere with. And you could add in like a cheap, I think they're about $200, a cheap 7-inch touchscreen, or you could just use your phone or your tablet or your laptop. And so, that's really exciting. But yeah, this repo has everything, including various troubleshooting and optimization and some other stuff that's in the pipelines. And there's various scripts and things in there to help you get up and running. More unboxing and setup notes. So that's more detailed setup and actually just getting the Xaviers up and running from a software perspective. And then this is that gist that I mentioned that's got some benchmarking, and it started as a benchmarking kind of platform, and then it turned into that amazing international collaboration with, I think there's over probably 100 odd posts in there now. So it is worth a read if you are interested. Documentation. So I've started to actually write down, I've had a lot of thoughts about various things. So I've started to write down, if you're looking at getting into Nanopore, or if you're in Nanopore and not using GPUs or wanting to invest in compute, I've written a bit of an article on, for whatever needs, what kind of compute plus GPU you need for various Nanopore sequencing tasks. And then getting set up on Linux machines has been a bit of a bottleneck. So I've just recently, a couple of days ago, written some documentation up on that. And then various presentations that I've given, including, not including the one I'm currently talking about, but others are all accessible there as well. And that just reiterates, if you're interested in GPUs and genomics, it's been pretty well received, and I think hopefully rather a helpful document. I've mentioned that GTC is happening next week. I'm talking, pre-recorded Miles from a week and a half ago is talking on the 13th, and present Miles has to be, or future Miles has to be there to answer questions in the chat at some ungodly. Oh no, it's not bad. It's 11 a.m. Okay, I thought it was some we are in the States. But so again, they're free to register, and there is a slew. I think there's 1,300 talks. But if you have your search terms of genomics or healthcare and things, you can get down to some really interesting information of what you are interested in. So parting thoughts. I'm really excited. I am really excited by the prospects of getting this out there and hearing people's thoughts and what they can do with it. I'm really excited in terms of the partnership that we have with iwi and other community members and things, of where we can take this and where other people can take it. Like, I might be being fanciful and I might be being naive because I'm a human geneticist, but I don't see it being such a stretch to having mobile labs in terms of maybe containers with fume hoods in them, where you could do extractions and then have these devices doing sequencing to monitor pandemic, or having these setups that can go out to like hospitals or out to ships and things really cheaply, and you have the power to do that. As teaching aids in school, I know Mick's got a couple of the nanos and is doing some really cool stuff, and we're in cahoots about doing some teaching work using these tools, because they are so cheap and so fast in terms of real time that you can really show students what genomics, or where genomics currently is and where it's heading potentially. I think back to when I was doing my undergrad and learning about NGS and pyrosequencing and and things like that, and it was cool, but and I don't know if I'm just again fanboying, but this just seems so much cooler, and the utility just seems huge. So yeah, again, I'm just starting to ramble, but I'm very excited, and I'm excited to hear what people think, and I'm excited to hear, like, I know there's a lot of people using this tech, and I know there's some really cool stuff out there. So people, I'll just acknowledge people that I get to spend my day talking tech and nerding out with, and I've mentioned Leah in terms of the clinical genomics work. She's been incredible. She's doing some amazing stuff. And I will reiterate, her documentation is immaculate. So if you want to go and see good documentation, or you want to go and play with the pipelines, you will have really good support in terms of that documentation. So do check that out. And Matt is awesome. He's a unicorn. He's great in the lab, and he's an amazing bioinformatician. So it's been awesome, working on this stuff with him. And obviously having you on board is awesome and incredible. And Donya's amazing. And Georgia, and everybody. It's just been a really great process and a really great team. So on that note, I think I will wrap up and have a bit of time for questions, Peter.